Everybody, it's Mrs. Peachy, and I'm going to do this video tutorial today on cell transport and how cells use this to maintain homeostasis. So first let's talk a little bit about what homeostasis is. Homeostasis it really is a state of equilibrium. So it's important for the cell to be able to maintain this state of equilibrium in order to survive. And all the time in your body you have, millions and millions of cells in there that are working to maintain that state of homeostasis so that you as an organism can maintain homeostasis. Some examples would be having like the ideal blood pH. It can't be too acidic, it can't be too basic, or that would throw your system out of whack. Having the ideal amount of blood sugar. If your sugars are too high or too low, then your body is not maintaining homeostasis. And if you know anybody who suffers from diabetes, you will know that they find it a challenge to maintain that homeostasis, and there are some significant health effects if their blood sugars become too high or too low. So those are just a couple of examples. There are many, many examples, but cells do this on an individual basis by using different methods to bring stuff in and to get stuff back out of the cell. And that would include like nutrients and ions and things like that to bring into the cell, but also to package and release all the waste products out of the cell so it doesn't poison itself. Okay, so we talked about homeostasis a little bit. Let's move on from there. And real quick, I want to go into why cells are small. Cells are small so that they can have the maximum surface area to be able to get as much stuff out and as much stuff in as they need to be able to survive. If they were very, very large and had kind of a smaller surface area, so that's that outside area that's, out, like the outside of your house, the walls of your house and the ceiling and the floor, that's your surface area. There wouldn't be enough exterior surface to get stuff out of the cell, or to be able to get enough stuff into the cell to make sure the cell maintains homeostasis. So cells are small. They want to have a maximum surface area to volume. So as little volume with as much surface area as possible. Okay, so we're going to talk a little bit about passive transport. Passive transport means that the cell does not need extra energy for this to happen. It just happens kind of on its own. It's like, an example I use is like a ball rolling down a hill, right? A ball rolls down a hill. It's going to do this, I'm going to give you a little picture here, without any real help. So if you put a ball at the top of a steep gradient, it's going to roll down the hill because it has what we call potential energy at the top of the hill, and it converts that to energy of motion, to that kinetic energy, and it moves downhill. A hill is sometimes called a gradient, and so that's a gradient of height. One end is high and the other end is low. When we look at how stuff moves in and out of the cell, this is another type of gradient. It's where one side of the cell is has higher concentration of something than the other side of the cell does. So things will move across the cell membrane from a high concentration to the low concentration in order to even that out. So the concentrations come to a state of equilibrium. And this is called diffusion. Diffusion is when particles move across the semi-permeable membrane of the cell membrane from an area of high concentration to low concentration. And this, again, goes on until a state of equilibrium is met. And then the particles can still move across the membrane in either direction, but the concentration on either side stays the same. So just a picture here kind of illustrating that. You have a higher concentration on the left side of this membrane, and then on the right side, say this is, for example, very concentrated salt water. And on this side, it is very fresh water. Well, the salt will move across the gradient until the concentration is the same on both sides of the gradient. Salt isn't the best example because in that case you would actually end up with water movement instead of the particles that move. But, you know, that's just one example of how this could work. Again, this is driven entirely by the motion of the molecules and the gradient itself. Okay, so it's just a matter of having more energy on one side, more molecules to move, than on the other side. Another example of a type of diffusion is osmosis. And osmosis is specifically the movement of water across a semi-permeable membrane, and it's just a little different than simple diffusion because it has to do with equaling the concentration of the solutions versus the number of molecules. Okay, so a lot of times when you make a solution, the particles in the solution are going to dissolve, or they'll, you know, basically they'll come apart in solution. And in this case, you'll have different concentrations on both sides of the cell membrane. So this says concentrated sugar solution, and this is dilute sugar solution. I like to think of this as very, very sweet Kool-Aid, and over here it's not so sweet. If you separated them by a semi-permeable membrane, then the water is going to move across, and the water is going to attempt to dilute this concentrated solution so that its concentration, its sweetness, is going to be the same on both sides of the membrane. So it's not the molecules that move in this case, it's actually the water that's moving. And that's because these sugar molecules are too big to be able to actually move across the membrane. So the water flows across, and look, if you look here, you do not have equal size volumes on both sides of this gradient here, but the concentration is the same. And by concentration, remember that, think of that as how sweet it is in this case, right? So these might have a total more sugar molecules, but they also have more water molecules, and these have less sugar molecules, but they also have less water molecules. But the ratio of sugar to water is the same on both sides. Alright, so here we come up with three terms, and vocabulary is a huge part of science, and specifically biology has tons and tons of vocabulary that is specific to biology. So the words we have here are hypotonic, hypertonic, and isotonic. And what I want you to become familiar with are the prefixes. Hypo, hyper, iso. Okay? Many of you know about hyperactive, right? So you have attention deficit hyperactive disorder, or ADHD. And you know that hyper means you're really, really energetic and you've got way too much energy, or you're just way more motion than you should have. So in this case, hyper always means more of something, more than is normal. Hypo means less than is normal. So like hypo, some people are hypothyroid. That means they're not producing, their thyroid is underactive, right? So that makes them sluggish and slow, and sometimes they gain weight because of it. So hypo is more, or excuse me, hypo is less, hyper is more, and iso means the same. So like an isosceles triangle. Isosceles triangles have two equal sides, right? So iso always means equal. So if you could remember those three prefixes, it's going to help you with a lot of things. We'll talk about hyper and hypothyroid at some point in biology, and other types of conditions that are hyper or hypo. And isotonic or iso, you'll use that prefix too. Those are all very common. So hypo means the concentration outside is lower than inside. Hyper means the concentration is greater outside the cell than inside. And isotonic is what will happen when they reach a state of equilibrium, because at that point they're the same. So here's some examples of what will happen. And again, just use common sense here. I don't think there's any need to memorize this. If the concentration outside the solution is greater than inside the solution, the water is going to flow out of the cell to try to dilute the outside of the cell. If the concentration is greater inside the cell than outside, the water will flow into the cell in an attempt to dilute it. And if it's the same, then water just flows in and out of the cell at the same rate. Remember, with osmosis, it's always movement of water with the attempt of diluting the higher concentration. Okay, and that's all passive transport. There are other methods of passive transport. One of them is called facilitated diffusion. And facilitated diffusion is something that is kind of assisted with these carrier proteins that are embedded in the cell membrane. So these proteins kind of act as a lock, and they have a specific shape to them. Then the molecules can actually fit inside the lock, which will open the protein and allow the molecules to pass through. This is a very much more discriminatory process. It's much more specific, and it helps with molecules that are just too big to fit through the cellular membrane. And an ion channel is similar to a protein channel. So in this case, there's again a specific ions that can fit in those channels, which are open, oftentimes gateways, to be able to move stuff in and out of the cell. Much later, we're going to talk about the sodium-potassium ion channel. That won't happen for quite some time. It's in second semester. All right. Real quickly, active transport. So passive transport does not require any extra energy. Active transport does. So it requires the cell to use ATP, which is basically the gasoline of the cell, right, of the body. It actually is what powers the whole body. It uses ATP to move stuff against the concentration gradient. So again, it has these proteins that are embedded in the cell membrane, and the proteins act to pump or kind of move things up against the gradient. So you can think of it like a water slide, right? In order to get the water up to the top of the water slide, you need to have a pump, and that moves the water to the top of the water slide for it to be able to move against the gradient. So you have to have something to move that water uphill. It's not going to flow that way on its own. When it goes downhill, that's due to just natural differences in energy. That's like diffusion. But when you're pumping the water up the hill, that is active transport. Alright, so another form of active transport is exo or endocytosis. And we talked a little bit about this, how the Golgi bodies are involved in this in some ways in our cells. But other organisms that are single-celled, they use this method to be able to eat. Amoeba, for example, use endocytosis to be able to eat, and it's actually called phagocytosis at that point. They take their whole cell membrane, just kind of comes out and surrounds the food particle, and then breaks off and forms like a circle, a little vacuole, and then they brings that whole thing into the cell and digests it with enzymes. So that's one method. And you can think of these pretty easy. Exo means you're bringing stuff out of the cell, and endo means you're bringing stuff into the cell. And in both cases, it's part of the cell membrane that's actually surrounding this, whatever it is that you're trying to move, and it forms what's called a vesicle. And remember, when you're moving stuff out of the cell, that vesicle is often made at the Golgi apparatus. That's what the whole purpose of that organelle is. Okay, so we just see a couple examples here of those vesicles. And that wraps up cell transport. So I hope you guys learned something today.